Hi, everyone. Welcome back to the CACNA1A Calcium Ion Channel. I'm Pangkong Fox, and I'm the Science Engagement Director for the CACNA1A Foundation. This is episode 3 of my science recap. So today's actually going to be less of a recap and more of a conversation that I want to start with all of you wonderful families out there, learning about how we can support the foundation in helping to identify and develop treatments that are going to work the best for our CACNA1A community. And this all starts with patient data. So the thing is, anytime we are approached by a biotech company or a pharma company, one of the first questions that they ask is, how many patients do you have? And so it's really important that we have a way to accurately identify and count how many patients are out there because these companies want to know what type of impact they're going to have and obviously what type of financial impact our patients are going to have on them. So we can identify patients, we can figure out what treatments are important by asking our patients directly through things like a natural history study, which is something that these companies often also ask. Do you have a natural history study? Do you have tools like patient-derived cell lines? Do you have animal models? A lot of these companies are looking for a disease that has a lot of information and some work done already and readily available tools. And this is what we mean when we talk about de-risking our disease. We want to make sure that when we that we can hand our disease over to these companies on a silver platter with a lot of the homework done, so that if they decide to partner with us, they are going to be ready to just jump right into the therapeutic development and the therapeutic design. And our pathway to clinical trials is going to be easier if we have a company that's backing us up. So let's talk about what that pathway really looks like and where patient data comes into play. Okay, so this is going to be a pretty simplified version of the pathway to clinical trials that we've mapped out at the foundation. And just think about it as how we're trying to get from point A, a rare disease, all the way to point B, our clinical trials. So what we need to do is build this bridge to span this great divide. And before we build anything, we need the right tools and we need the right foundation on which we can build that. So the first thing that we need to do is to create a preclinical toolbox, put all the tools that are needed to start building that foundation of knowledge. And that comes from disease research. So we start with basic research, which allows scientists to go into cells and study what's going on, study those nitty-gritty details of the disease mechanisms, how does each variant contribute to the disease? And that leads into therapeutic development. So this is translational research where we're taking the data from basic research and turning it into something that can be used on patients. And that feeds directly into proof of concept studies. And these are the experiments that are really important because they show that the disease, the gene, that these are amenable to treatments. And that's what the FDA likes to hear. And of course, there are going to be times when we have to circle back to the basic research, go back and develop something because not everything is going to work right away. But once we've identified that therapeutic candidate, we then go and file something called an IND application with the FDA. And the IND stands for the Investigational New Drug Application. And if that is then approved, then that drug or that treatment will go into clinical trials. And so if we're talking about attracting an industrial or an industry partner, we're hoping that they can jump in anywhere here, maybe right away in the beginning, if we have a strong toolbox, or anywhere in disease research, therapeutic development, and proof of concept studies. So there's a lot of work to do. And obviously, we need patient data as well as engagement, because when it comes to clinical trials, we will also need a patient population that's ready to enroll in order to assess whether that treatment is working or not. And we need money. So again, that's why we've been focusing on fundraising these last couple of years so that we can make sure that this important research does happen. So how are we currently using patient data to help de-risk our disease? Well, we're using it to build our preclinical toolbox. So data collection programs and platforms such as our natural history study, our data collection program with RareX, these are providing important information like the number of patients, the variants out there, symptoms, treatment gaps, and we're going to be expanding those efforts with Citizen that we'll be launching in the spring, and our disease concept model study that we'll be launching in the summer. Our biobanking program currently has 15 patient samples with 3 completed iPSC lines and and 5 more in progress. And remember that these are the cells that can be induced and turned into any type of neuron, which creates a really great environment for scientists to study what these variants and what this disease is doing outside of the body. Same thing with these animal disease models that are carrying human pathogenic variants. And then we will also be launching a consortium effort to identify biomarkers. And this is important because we will be looking for biological signatures or traits that are common to CACNA1A patients. And this will really help with patient diagnosis and identification, as well as clinical trial design. Because biomarkers can actually be used as a way to assess whether a treatment is working or not. And finally, I just want to end with this figure that I put together here, and it's summarizing, what we can accomplish or achieve using patient data. This data is the foundation of all the work needed to get to clinical trials, and this is why it's so important for CACNA1A families to engage with the foundation and enroll in the studies so that we can make our way to getting treatments approved for our CACNA1A community. Okay, so I hope that that was helpful in understanding why patient data is so important, especially for our rare disease community. So remember, patient data helps us de risk the disease by providing information about patients, symptoms, what types of treatments could work. It helps us build a strong preclinical toolbox, and it's also going to help us attract strong partners from industry as well as from academia. There are lots of researchers out there at different universities and different institutions who are working really hard to help our CACNA1A community. So we need as much help as we can get here to get us through to clinical trials. So remember to sign up for our natural history study. Remember, this is being run by Dr. Wendy Chung of Columbia University. She is a world-renowned medical geneticist. We are so fortunate to have her partnering with us and heading this for us. And don't forget to sign up for our data collection program through RareX. Both of these programs are going to put our data sets in front of many different eyes, and that's only going to be to our advantage. If you have any questions about either of these programs, information is provided on our website. So I'll stop here, and I will be back in February with some more science recaps.